Greetings everyone and welcome to another exciting genetic engineering and biotechnology news webinar. Our presentation today is entitled Addressing the Challenges of Therapeutic Vector Development. I'm Jeff Bogaliskas, technical editor for GEN, and I'll be the moderator for today's webinar presentation. Investigators have long understood that gene therapy is as good as the vectors that are utilized to deliver the therapeutic payload. However, this knowledge hasn't made vector development any less labor-intensive, with many researchers not giving much thought to how the development process may affect important downstream applications. Yet often when development strategies become over-complex, innovation steps in to unburden methodologies and streamline research workflows. Let's meet our speakers for this GEN webinar who will discuss solutions to aid vector creation, address factors that are critical for viral vector development from analytical to large-scale manufacturing settings, and inform us about recent gene therapy successes. Miguel Dominguez is a global distribution manager at Mirus Bio. Mr. Dominguez will briefly describe some of the recent solutions Mirus has generated for lentiviral vector development that could significantly improve research endeavors. Johannes van der Loo is the director of the Clinical Vector Core at Children's Hospital of Philadelphia. Dr. van der Loo will discuss several approaches for the scale-up of viral vectors for use in clinical therapeutics. Boris Kantor is an assistant professor and viral vector core director at Duke University. Dr. Kantor will address some of the challenges that investigators face when creating lentiviral vectors and some of the recent progress made for gene therapy studies. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply hit the Ask a Question box below the presentation screen, type in your question, and hit OK. All right, let's begin our webinar and let the information flow. Miguel, take it away.
Hi, and good morning. Thanks for that introduction. My name is Miguel Dominguez, and I'm Global Distribution Manager for Mirus Bio. I'll take only a few moments of your time to introduce you to Mirus and tell you about our offering for high-titer lentivirus production. For over 20 years, Mirus Bio has been a pioneer in nucleic acid delivery solutions to support both molecular and cell biology applications to give researchers unprecedented control of the genome at their fingertips. Our offering spans three major nucleic acid delivery platforms that include chemical transfection, electroporation, and more recently, viral transduction. Our high-quality solutions, along with a robust technical support, allow us to play a role in programming the genome in order to provide scientists with turnkey tools to add, delete, or modify any gene at will. This all ultimately underscores our commitment to support any delivery method capable of delivering any nucleic acid into any cell type. Now, with this brief introduction to Mirus, I want to tell you about our most recent product introduction relevant to the webinar topic today, the transient lenti transfection reagent. This novel reagent supports high-titer functional lentivirus production and is a solution designed to enhance delivery of lentiviral vectors into both adherent and suspension HEC293 cell types to provide significantly higher functional titers, which can eliminate timely concentration steps and allow for more robust transductions downstream. Our testing also shows higher functional lentiviral titers produced with transient lenti over other formats. Here we show functional lentiviral titers measured by GFP expression. Lentivirus was produced using transient lenti, lipofectamine 2000 or 3000, linear PI, and calcium phosphate to deliver the Sigma lentivirus packaging mix powered by mission vectors. Lentivirus was harvested, filtered, and titered in HEC293 T17 cells. You can clearly see transient lenti yields higher fold production of the lipofectamine reagents and even higher titers over PI and calcium phosphate. Then with higher functional titers, one can also expect better transductions downstream. Here we show better transduction efficiency in eye-cell motor neurons from Cellular Dynamics International. These are a model for primary motor neurons which are very difficult to transfect. We used equal volumes of unconcentrated lentivirus produced using either transient lenti or lipofectamine 2000 for transductions. As you can clearly see, we're able to achieve 5-fold higher infection efficiency with lentivirus produced using transient lenti than with virus produced using lipofectamine 2000. Higher lentivirus titers therefore allows for better transduction optimization of your target cell. And in addition to our own internal testing, we received very positive feedback from several researchers, like the following data from a viral vector core lab. They compared virus production between calcium phosphate, lipofectamine 2000, and transient lenti. Their results showed such an improvement in production that they have now incorporated the transient lenti transfection reagent within their regular workflow. You can also find more information on transient lenti transfection reagent, including data, protocols, and how to sample by visiting our website at www.mirasbio.com/lentivirus. Additionally, tomorrow we also launch a new solution, Transient VirusGen, for high-titer AAV production in both adherent and suspension HEC293 cell types. Please visit our homepage to find out more and to request samples of the new product. Thanks for your time, and please enjoy the presentations from Dr. Vanderloo and Dr. Cantor.
Thanks for getting things rolling, Miguel, and for telling us about some of the excellent vector development solutions that MIRAS has to offer. Before we move on to the next presentation, I want to remind the audience to submit questions for our Q&A session at the end of the talks. We'll try to answer as many questions as we can, so simply hit the Ask a Question button below the presentation screen, type in your question, and hit OK. All right, Han, the audience is tuned in and waiting to hear what you have to say.
Thank you for the introduction. Today we'll focus on large-scale vector manufacturing, and specifically the use of transient transfection methods and stable producer cell lines. It has been 27 years since the first gene therapy trial was conducted on a patient with ADA deficiency. And in those 27 years, the field has seen a dramatic resurgence. There are in total worldwide over 2,400 clinical trials active at this time, and the targets include the eyes for diseases such as Leber's congenital amaurosis, glaucoma, retinitis pigmentosa, skeletal muscle and heart for muscular dystrophy, the lungs for cystic fibrosis, the liver, for instance, for storage diseases, the gastrointestinal system, blood for the treatment of cancer with immunotherapy, the treatment of sickle cell disease and thalassemias, as well as the skin. There are two types of gene therapy approaches. The first one, the traditional approach, is called gene addition and involves the addition of nucleotide base pairs, a single gene or multiple genes, to the genome of a cell. This is used in situations where a single gene defect results in a loss of function, and addition of the correct copy of the gene that does not have the defect then restores the deficiency and therefore cures the disease. As you can see here on the right-hand side, there's an example of an adenoviral vector providing a copy of the corrected gene to the cell, and the corrected gene can maintain in the cell either episomally, meaning non-integrated, or integrated into the genome. The second type of gene therapy is called genome editing. That includes insertion, deletion, or replacement of DNA using engineered nucleases. The nucleases provide site-specific double-strand breaks in the DNA that is then repaired through non-homologous end joining or homologous recombination. There are 4 types of nucleases: the meganucleases, zinc finger nucleases, TALENs, which stands for transcription activator-like effector-based nucleases, and the CRISPR-Cas9 system, or the clustered regularly interspaced short palindromic repeats. A separate category of genome editing is base editing, where single nucleotide changes are made that do not require the creation of a double-strand break. There are 2 ways genes can be delivered to the patient. Either by direct delivery, where the therapeutic gene is packaged into a delivery vehicle such as a virus and then injected into the patient directly, either into a target organ or IV. The other way is a cell-based delivery, and as is shown here, stem cells are isolated from the patient, they're propagated in the laboratory, and then exposed to the delivery vehicle that includes a therapeutic gene. The cells that are genetically modified are then reintroduced into the patient. The first method is called in vivo gene therapy. The second method is called ex vivo gene therapy. Frequently, the delivery method the vector is a virus, since that is what viruses do best, being developed through evolution over 3.5 billion years. These are the four most commonly used viral vectors: the adenovirus, adeno-associated virus, retrovirus or gamma retrovirus, and lentivirus. They differ in several ways, including the size of their genome and hence their packaging capacity for delivering therapeutic genes. Adenovirus and adeno-associated virus are known as non-integrating vectors that target non-dividing cells, or can be used to target non-dividing cells such as brain, muscle, and liver. The gamma retrovirus and lentivirus, on the other hand, are known as integrating vectors to target dividing cells, including stem cells and CAR T cells. The integrating vectors are needed for transducing cell populations that divide and expand. And the reason for that is that any transgene that is maintained episomally is not duplicated upon cell division, and so the message gets lost when the cells divide and expand, whereas integrating transgenes are copied when the genome is copied and appears unchanged into the daughter cells. A special type of lentiviral vector is the non-integrating integrase deficient vectors, and you will hear in the next talk from Dr. Cantor, who will elaborate on the integrase deficient lentiviral vectors. So vector tropism is defined by the proteins with which the virus contacts the cell. And the tropism for adenovirus, for instance, can be modified by changing the fiber knob. Lentivirus and also gamma retrovirus, it can be pseudotyped by changing the envelope of glycoprotein, and then the adeno-associated virus can be targeted by changing the type of capsid. And as you can see here, for the AAV serotypes 1 through 9 in the mouse, different serotypes target different organs when the virus is injected intravenously. So how are viral vectors being generated? As illustrated here for gamma retroviral vectors, the genome is divided into packaging and vector components. So first let's look at the top of the diagram. This is a wild-type gamma retrovirus. On the left-hand side in red, the LTR, or long terminal repeat, and then the structural genes GAG and PAL, as well as the envelope gene, are being depicted here in yellow and blue. Psi, the packaging sequence, is located between the LTR and the GAG, and Psi is responsible for pulling the strand of DNA into the viral particle when the virus is being packaged. So the packaging plasmid generally then contains the GAG, PAL, and envelope genes. Frequently this is presented to the cell on separate plasmids, one for the VSVG and one for the GAG, PAL. On the other hand, there's the retroviral vector, which now has a blank space between the LTRs, and the transgene, the gene of interest, is inserted into that space, cloned into that space, so that when a viral particle is being assembled, this is the only piece of DNA that has the packaging sequence included, and therefore this is the only piece of DNA that will then get assembled into the viral particle. The packaging plasmids with the GAG, PAL, and envelope proteins are present in the producer cell but don't get packaged into the viral vector, and therefore the vector itself is replication deficient upon infecting another cell. The cell is transduced, but no additional virus is being made. The three manufacturing methods that I want to briefly touch on today are transient transfection, stable producer line, and the baculovirus SF9 insect cell system, which is specific to AAV. In transient transfection, cells are transfected using the plasmids that carry all the genes necessary to produce a virus. In a stable producer cell line, all the genes have already been stably incorporated, and anytime the cells are being cultured, the virus is being produced. And in the baculovirus system, instead of transfecting cells with the genes that are necessary to create viral particles, the cells are infected using baculovirus. And the baculovirus carries the genes necessary to create, in this case, AAV. In the transient transfection, HEK293 cells are transfected with the different plasmids that are needed to create the viral particles, the vector plasmid, the GAG-POL, and the envelope plasmid. This is an example of gamma retroviral generation. Then over a period of 2 to 3 days, the viral proteins are being expressed and particles are being assembled and released into the media. The benefits of transient transfection is that the manufacturing process is relatively fast. It also allows for last-minute changes when the investigator wants to make a change in the vector plasmid or wants to change the serotype of the vector. The only thing that needs to change is the plasmid that is being used, while the same cell bank is available for production. And then the other benefit is there's only one certified master cell bank needed for all products. Challenges are that the cells produce virus only using a narrow time window, so it needs to be carefully timed. The transfection process itself is pH, temperature, and time dependent, and frequently also operator dependent, which of course depends on the training of the operator, and transfection is challenging to standardize and scale up. An alternative way of producing virus is through the use of a stable producer cell line. The principle is that a cell line is created that includes all the genes necessary to form viral particles. And in order to do this, it's usually a two-step process where the packaging plasmid is first introduced into the packaging cell line and the cells are selected for those that express the packaging sequences. And then a second selection where the cell line is transfected with the vector plasmid. The cell line is then banked, certified, and at that point, anytime the cells are being thawed, the cells will produce virus. So the main benefit is that there is a continuous production of virus when growing the cells. The cells have been selected for high productivity, and the system is easy to scale up. The challenges here are that the packaging proteins or even the transgene may be toxic to the cells and require an inducible promoter. So that complicates the generation of the cell line. Secondly, you will require a certified master cell bank for each product. And finally, the genetic stability of the selected clone is not necessarily guaranteed and need to be carefully evaluated prior to using the cells. The third system is the baculovirus system that utilizes SF9 insect cells. And the principle here is that cells are not transfected with plasmid, but are infected using baculovirus that carries the genes needed to create viral particles. The benefits are that the insect cells are grown in suspension culture, the system is easily scalable, and the cells are easily infected. The baculovirus also is non-infectious to humans. The challenges with the system are that baculovirus needs to be ultimately removed from the clinical product, that you require 2 certified baculovirus viral banks for each product. All the genes that are necessary to create viral particles cannot be put in a single baculovirus, so a second baculovirus is needed to get all the genes on board. And then finally, the ratio of the 2 baculoviruses and the timing of the harvest are critical. As an example, here on the left-hand side at the top you see SF9 cells that have not been infected with baculovirus, and on the bottom is a culture that has been infected with baculovirus. And the timing of harvest for AAV can be estimated based on the cytopathic effect. You see here that the cells in general are bigger and swollen and ready to be lysed, by the way, with baculovirus. The timelines for transient transfection and stable producer cell lines are quite different. In this diagram depicted here on the horizontal axis, you see the number of months that it takes. In blue, the amount of time needed for cell culture, in green for virus production, and in red for certification and QA release. As you can see, transient transfection takes 4 to 5 months, where the cells are expanded and then transfected. Virus is produced over a short period of time, and then there is the 3 to 4 month period of certification and release. Stable producer cell lines, on the other hand, the cell expansion is much more elaborate. It requires a first round and a second round of selection for the different genes, cloning of the cell line, and then followed by certification. Once this master cell bank, a specific master cell bank for each product, is then certified, the cells are thawed, virus is produced over a period that is longer, as can be achieved with transient transfection. For over several weeks, a product is collected then, and then the product is certified and released by QA. So in total, that is a process that takes 13 to 14 months. Once the first product has been manufactured, then each subsequent product will take less time, very similar to transient transfection, about 4 to 5 months for each. So to summarize the first part, the transient transfection is well suited for phase 1-2 studies because it allows last-minute changes to vector and packaging plasmids. The amount of material achievable at volumes up to 200 liters is sufficient for most early phase studies. The yield can be increased using scale-out, which is achieved by increasing the number of batches manufactured. The approach has an attractive manufacturing timeline of 4 to 5 months. A single lot manufacturing is price competitive as compared to the use of stable producer cell lines, and it only requires one common certified master cell bank. The stable producer cell line, on the other hand, is well suited for phase 3 and commercial manufacturing. At this point, the vector construct is well defined, is no longer going to change. The approach allows for true scale-up to larger volumes, not just scale-out. The amount manufactured per lot can be larger, and after a master cell bank has been created, the manufacturing is completed and product is certified in about 4 to 5 months. And ultimately, pricing is favorable when manufacturing multiple lots. In the next part of the talk, I want to focus on adherent and non-adherent cells. And as illustrated here in the diagram, a cell that is non-adherent is inherently different from a cell that is adherent. And you can see that when a cell is non-adherent and goes to adherent conditions, there is actin polymerization, there are stress fibers involved in the attachment process and the spreading out of the cells on the surface. And this has consequences for the biology of the cells and therefore for the ability of cells to produce virus. So let's look at both adherent and non-adherent cell cultures, the benefits, the limitations, as well as the systems that can be utilized to grow cells. The main benefit of adherent cell culture is that the viral titers are generally higher as compared to suspension cells. Among the limitations are that the cell growth is limited by the surface area. Although the capacity can be increased using scale-out, it is very hard to do true scale-up with adherent cell cultures. Scale-out systems require additional effort and they increase risk. In a packed bioreactor, the cell population may not be homogeneous, the cells cannot be harvested, and collecting intracellular virus requires chemical lysis. Compatible systems with adherent cell culture are the culture plastics, such as flasks, roller bottles, stacks, hyperflasks, hyperstacks. The scale-up version of that would be the packed or fixed bed bioreactor. Several types are listed here: the cell cube, the iCELLis, the Cellogen Plus bioreactor, the wave bioreactor that includes a carrier on which the cells grow. In this particular case, I mentioned fibrocell as the carrier, and the stirred tank bioreactor that utilizes microbeads for the adherent cells to grow on. Suspension culture, on the other hand, the benefits are that suspension cultures are compatible with true scale-up. The cells are easily passaged, the cell population is homogeneous, the cells can be efficiently transfected, Both cells and media are easily to harvest, and one of the other benefits is that these cultures can be serum-free. The limitations of suspension-based cultures are that they require a suspension-adapted cell line. The cell growth is limited by the concentration of the cells, not necessarily not the surface area on which they grow, but the concentration. And viral titers tend to be lower as compared to adherent cells, which may ultimately impact product purity. Among the systems that are compatible with suspension cell culture, I listed here the stirred tank bioreactor, the wave bioreactor, and the packed or fixed bed bioreactor, which can be used for suspension cells as well. And in this particular case, for instance, for iCellis, the cells are trapped in the matrix and produce virus in the fixed bed reactor. So ultimately, the choice of manufacturing system depends on the type of viral vector, the availability of the cell lines for high yield production, and the amount needed in clinical phase. Some sponsors that we talk to prefer to choose a manufacturing system compatible with both early trials and commercial manufacturing, which means that they are starting with the end in mind versus switching horses midstream and having to do compatibility testing when switching manufacturing method. Common constraints in the choice of the manufacturing system generally are of financial nature or have to do with the timeline. When using transfection for large-scale manufacturing, it is very important that the information that you have on the plasmid is complete. That the plasmid names that are being used on the vial that you receive, the sequence that you receive, the map and the paperwork, that they all match, that the actual sequence matches the predicted sequence. It is also important to make sure that the plasmid is homogeneous and that it is stable during amplification. That the transient plasmid does not exceed the vector packaging capacity. Also that the backbone is sized to prevent co-packaging of genomic or plasmid DNA sequences. For instance, the AAV particle is known to co-package sequences, or frequently co-package sequences when the backbone is not sized adequately and there's too much space, and the viral particle will basically just pull everything in until it is filled. Make sure the potency of the vector has been tested, that the plasmid is in the correct buffer and the correct pH, and simply that it is the correct plasmid. The due diligence when working with sponsored provided plasmid at large scale includes proper documentation, meaning you need an annotated plasmid map, plasmid sequence, and growth information to amplify the plasmid. When amplifying the plasmid through a service provider, make sure that you audit the service provider, have a quality and a services agreement with the provider, and negotiate specifications and in some cases custom specifications. And finally, perform quality control. And at a minimum, include visual inspection, testing of bioburden and endotoxin, determine the purity and concentration, the pH, and last but not least, sequence the entire plasmid before you go forward. Finally, a few words about the choice of reagents and the process when performing transfection, especially at large scale. Make sure that you choose reagents that have the highest purity that is available. Use a transfection reagent that is compatible with the target cells and doesn't affect target cell viability. Use low endotoxin plasmids that have been fully sequenced. Regarding the process, keep in mind that the procedure may not scale. For instance, mixing of reagents at a 1 ml scale is not identical to mixing reagents at a 10 liter or 100 liter or more or larger scale. Be sure to control the temperature, be sure to control the time, and thoroughly train your personnel. Then in summary, we talked about gene therapy in general, targets and diseases, the difference between gene addition and gene editing, direct and cell-based gene delivery. We spoke about integrating and non-integrating vectors and vector tropism. The second, I highlighted three production methods for viral vectors: transient, transfection, stable producer cell lines, and the baculovirus system. We talked about large-scale culture systems for adherent cells and non-adherent or suspension cell cultures. And finally, we talked about transient transfection, challenges, and due diligence. On this slide is my contact information, so feel free to call me or send me an email if there are additional questions after this webinar is complete. And with that, I would like to give it back to the organizer. Thank you.
Thanks, Han, for that informative presentation. I think our audience now has a greater sense of the numerous considerations that need to be examined when planning to move to large-scale vector manufacturing. Before we move on to the last presentation, I want to remind the audience once again to submit questions for our Q&A session at the end of the presentations. We'll try to answer as many questions as we can, so simply hit the Ask a Question box below the presentation screen, type in your question, and hit OK. All right, with that, let's start our last presentation. Boris?
In the current presentation, we will focus on integrase-deficient lentiviral vectors, and here we'll address challenges and recent progress of this platform for gene delivery and gene therapy. Lentiviral vectors are one of the mainstay platforms for gene delivery and gene therapy because of the following advantages. They demonstrate low immunogenicity. They can carry large payloads up to 9 to 10 kilobase pairs. They demonstrate wide range of tropism, basically can transduce any possible cell or organ. They transduce very robustly both dividing and non-dividing cells, which separate this platform from, for example, simple retroviral vectors, which cannot transduce non-dividing cells. And they support a high level of stable and long-term expression. The major disadvantage of all integrating platforms, including retroviral vectors and lentiviral vectors, is the inherited risk of insertion mutagenesis. And very unfortunately, this risk materialized in clinical trial in which attempts were made to correct X-linked severe immunodeficiency syndrome, when several patients participated in this clinical trial, developed severe leukemia, and 2 patients died from severe leukemia, and it's been linked to integration of retroviral vector cassette carried therapeutic gene in proximity to oncogene called LMO2, which cause upregulation gene expression of LMO2 and the expansion of T cells. To circumvent this problem of insertional mutagenesis, we and others focus on development of non-integrating episomal lentiviral vectors. Non-integrating episomal lentiviral vectors can be generated through utilization of 2 approaches, 2 strategies. One is through altering ET sites located at 5' and 3' LTR on expression cassette, and those sites are scaffold binding sites for integrase protein. And it has been demonstrated that mutation in ET site may completely abrogate integration process. Second, mostly utilized in the lab method is through introduction of point mutation in catalytic domain of integrase protein, and it's been demonstrated that any of these mutations at position D64, D116, and E152 can completely abrogate the integration process. Now, enable of integration, this linear form which derived from RNA of the virus can remain in the nucleus or can be further converted into single LTR or double LTR through a homologous direct recombination mechanism or non-homologous end joining mechanism. Production of integrase-deficient lentiviral vectors is quite similar to production of integrase-competent lentiviral vectors. We use transient transfection protocol in which 3 plasmids, transfer plasmid, carry transgene of interest, packaging plasmid, harbored mutation in integrase open reading frame highlighted here as D64E, and envelope plasmid delivering envelope of interest, in this case vesicular stomatitis virus G, delivered into 293 T cells by transient transfection protocol. Mostly we utilize calcium phosphate protocol of transfection, and then 48 to 72 hours post-transfection, viral particles can be collected from the supernatant, applied directly on target cells, or can be further concentrated and purified to achieve higher production yields. The major advantage of integrase deficient lentiviral vectors stem from their lower ability for integration. Indeed, as we previously demonstrated, the rate of integration of ideal Vs is about 0.2%, which is one event per 500 transduced cells, which is significantly lower in comparison to ICLV integrase competent lentiviral vectors, in which we found rate of integration as high as 30%. The major caveat of integrase deficient lentiviral vectors stems from their low level of expression. And as we previously demonstrated, that regardless of promoters utilized to drive expression of reporter gene, in this case GFP, we found that integrase deficient lentiviral vectors show a significantly lower expression of GFP in comparison to integrase competent lentiviral vectors, here highlighted by a flow cytometry experiment in upper panel versus integrase deficient lentiviral vectors as highlighted in lower panel. We previously elucidated mechanism responsible for this low level of expression of IDLV genomes, and we linked it with formation of close condensed chromatin conformation around IDLV genomes, which we found to be incrustated with post-translation histone modification typical to silence and repressive chromatin organization, such as K9 DN2 methylation, deacetylation, other, which altogether form a close chromatin which is restrictive to RNA polymerase II and general transcription machinery. More importantly, we were able to demonstrate that this condensed chromatin organization formed around IDLV genomes can be reversed through targeting of repressory complexes such as, for example, histone deacetylases, which resulted in formation of new open chromatin organization around IDLVs typical to L-chromatin, which we showed to be incrustated with post-translation histone modification typical to active chromatin such as K4, DN2 methylation, acetylation, and others, which led to activation of IDLV genome transcription. Investigation and elucidation of factors playing a role in downregulation of IDLV genome transcription were instrumental for identification of cis-actin elements located at UC region of 3' LTR, highlighted here as a second generation of IDLV platform, and we demonstrated that deletion of this region may cause upregulation in gene expression of IDLV platform. If we compare between second generation and first generation of IDLV, we can see that the second generation expressed at a higher level. Interestingly, we didn't observe any upregulation in gene expression of integrase competent lentiviral vectors as highlighted here in upper panel. From the right side, there is a northern blot analysis which demonstrate again increase is in synthesis of messenger RNA of IDLV. Here is the second generation compared to significantly lower amount of RNA from the first generation of IDLV, and the left panel of northern blot image demonstrate a comparable amount of second and first generation of integrase competent lentiviral vectors messenger RNAs. Similar results were demonstrated when IDLV vectors were injected in striatum of rats. Here, left image, if we take a look on the lower panel, we can compare between second generation and first generation of expression of GFP from IDLV genomes. And again, consistently with previously demonstrated results, we didn't see an increase in a second generation of integrase competent lentiviral vectors. And right panel referring to the experiment in which we utilize liver-specific promoter to drive expression of firefly luciferase. And again, if we compare between second generation of IDLV expression to first generation of IDLV expression, we can see very significant increase in gene expression for the second generation of IDLV, which getting really close to actually to expression of integrase competent lentiviral vectors here highlighted in a second row from the left panel. To further enhance production and expression of integrase deficient lentiviral vectors, we next focus on transcription factor SP1, which present in multiple copies throughout HIV genome, and it has been previously demonstrated to play central role for HIV infection and pathogenesis. Indeed, deletion in SP1 binding site was shown to be able to abrogate HIV replication and virulence, and conversely, duplication of SP1 binding sites were shown to be able to transform weak and non-pathogenic HIV strains used for vaccination into highly virulent and pathogenic HIV. Importantly, SP1 binding sites are completely deleted from most of seen self-inactivating lentiviral vectors. To further develop integrase deficient lentiviral vectors, we focus on application which we thought would be highly benefited from the use of transient delivery platform. Here we focus on application involved gene editing system, specifically aiming to develop integrase deficient lentiviral vector platform for efficient CRISPR-Cas delivery. Now, as the time limited, I would not be able to expand on introduction of this gene editing platform. I would only mention that CRISPR-Cas evolved and adopted recently from bacteria, where it operating as adaptive immune system and has been extremely widely utilized for highly efficient gene editing manipulation, including gene editing, gene repression and activation, drug discovery, and many others. It should be noted that even though integrase competent lentiviral vectors are one of the main platforms for delivery of CRISPR-Cas, one of the major disadvantages of integrating vectors is the high risk of highly undesirable off-target effects. IDLV from this perspective, delivering CRISPR-Cas transiently would be highly beneficial. And second of all, as it has been demonstrated, lentiviral vectors developed so far for delivery all-in-one CRISPR-Cas cassette demonstrate low production titers. Indeed, as highlighted here in figure B, even the most advanced lentiviral vectors platform called CRISPR-V2, which has been developed in Dr. Feng Zhang's laboratory, demonstrate significantly lower titers in comparison with a binary system delivering gRNA component and Cas9 from two separate plasmids. Another limitation of CRISPR-V2 mentioned in the previous slide is that it's oversized plasmid. The size of V2 is more than 15 kilobase pairs, which generates significant difficulty for efficient propagation of this plasmid in bacteria and creates possibility for unwanted recombination events. In aiming to trim the size of this plasmid down, we deleted unnecessary buffer sequences highlighted in the upper panel, which reduced size of CRISPR-V2 by about 2 kilobase pairs. We also placed two BSMB1 sites used for cloning of gRNAs close to each other and created unique BSRG1 site for ensuring efficient screening of gRNA-positive colonies. The major improvement was achieved by cloning of two SP1 sites upstream from U6 promoter, as highlighted in the lower panel. We demonstrate here that by incorporation of two sp1 binding sites into the short version of CRISPR-Cas plasmid, we were able to increase quite significantly production titers of IDLV CRISPR-Cas vectors, as measured here by P24 ELISA, highlighted here as open white bars. Intensely, we also demonstrate similar effect on integrase competent lentiviral vectors, production of which was also enhanced by sp1 cloning. Significantly, we demonstrate that functional production titers of integrase competent lentiviral vectors delivering CRISPR-Cas as a mean of counting promycin positive clones was increased by 7 to 8-fold. We next wanted to test whether the novel IDLV CRISPR-Cas system can be used to achieve robust and safe gene editing manipulations. To this end, we created 293 T cells express GFP reporter, and we target these cells with IDLV or ICLV all-in-one CRISPR-Cas system. As highlighted here in flow cytometry experiment, we demonstrated very efficient knockout of GFP as early as 7 days post-transduction with both IDLV and ICLV vectors. We observed near to complete depletion of GFP signal for both platforms at 14 and 21 days post-transduction. Overall, we demonstrate here a highly efficient gene knockout in dividing cells with integrase deficient lentiviral vectors, which we found to be quite comparable to the integrated counterpart. To check if IDLV CRISPR-Cas can be used for efficient knockout in non-dividing cells, we transduce primary neurons with GFP reporter virus, or virus carried MCR reporter transgene. And the next day, we super transduce these cells with all-in-one IDLV vector, which harbored gRNA to target GFP. We demonstrate depletion of GFP signal, which was significantly improved by using vector which harbor SP1 repeat. We observed no reduction in MCR expression, which suggests high specificity of the system. Next, to validate the system in vivo, we injected SP1 IDLV vector carrying gRNA to target alpha-2 subunit of GABA receptor complex. The vector was injected into nucleus accumbens of the rats, and the efficiency of knockout was assessed by Western blot, highlighted here the upper right image, or by measuring inhibitory postsynaptic current. We demonstrated a complete reduction in protein appearance by Western blotting, and typical increase in variability of inhibitory synaptic currents as a result of alpha-2 subunit depletion. Overall, we demonstrate here that the novel IDLV vector can be used to achieve efficient gene knockouts in non-dividing cells in vitro and in vivo. To check the off-target capabilities of integrating and non-integrating platforms, we transduced 293 T cells with IDLV and ICLV vectors, delivering gRNAs to target GFP, and following transduction, executed whole exome sequencing. The analysis of the insertions and deletions in non-specific to GFP sequences reveals 17 genes in which CRISPR-Cas delivered by integrating vector induce significantly higher level of indel formation than integrase deficient vector. This observation clearly suggests that IDLV vector platform has safety advantage over integrase competent vector. To summarize this presentation, here we present the third generation of integrase deficient lentiviral vectors harbored large U3 deletion and SP1 insertion. We demonstrate that CRISPR-Cas delivered by the novel IDLVs is capable to achieve safe, rapid, and efficient gene editing in dividing cells and post-mitotic neurons. We believe that the novel IDLVs will be a great addition to the arsenal of non-integrating vectors for application requiring only transient delivery. Such application may include gene editing, human embryonic stem cells differentiation, immunization, cancer immunotherapy, and replacement therapy as a means of correction of monogenetic disorders. With this, I would like to conclude this presentation by acknowledging our collaborators from University of South Carolina, Dr. Pavel Artinsky and Benedito Donovan, sequencing facility at Duke University headed by Dr. David Corcoran, members of my lab, Shiram, Kate Illich, Megan, Roger, Lisa, and Malik. I would like to acknowledge that support for the study was provided through RDF and NIH grants. Special thanks to Jeffrey Boguliskis for organizing this seminar and for inviting me to give this presentation, and my co-presenters, Dr. Jones van der Loo and Dr. Miguel Dominguez. I would like to thank you all for listening to this presentation. Thank you very much.
Thanks, Boris. I think that was a really great presentation to end this webinar, especially with some really encouraging results for the future of gene therapy. Before we start the Q&A session, I want to let everyone know this is their final chance to submit questions for our speakers. Hurry up and send them in now. We'll try to get to as many questions as we can, so simply hit the Ask a Question button below the presentation screen, type in your question, and then hit OK. All right, it looks like we have a bunch of good questions that have already come in, so let's begin the Q&A. We'll try to get to as many questions as we can. Give us a few moments on our side to get everything situated, and we'll transition into the Q&A session presently. Let's begin the Q&A. We got a bunch of questions that have rolled in and some really great ones. So we're going to start off with our first question for Miguel. Miguel, one of our audience members would like to know, how does transfection efficiency of the packaging cell relate to functional virus titers?
Oh, hi, Jeff. Thanks. Yes, that's an excellent question, actually. We found that one may not necessarily be indicative of the other. You definitely want to start off with a high transfection efficiency, which is the case for our transient transfection reagent. However, during development and screening, particular formulations that may have led to a high transfection efficiency didn't always lead to high viral titers, which is one reason why we always strongly recommend doing a functional titer of your virus after transfection to make sure that it is of a high concentration. And in particular, when performing any side-by-side comparisons with an existing formulation that you might be using, definitely make sure that the output is measured through high functional titer. And again, that's something that we've uncovered time and time again with transient lenti, is that it does lead to high functional lentivirus titers. But great question. Thank you.
Thanks, Miguel. Han, our next question is for you. Audience member asks, is GMP plasmid needed for clinical vector production?
So, that's an interesting question, and we do get it a lot. For large-scale manufacturing, and the answer is really it depends on where the vector is going to be used. And also, in the case of, for instance, gene therapy in vivo, when plasmid is injected straight into the patient, that's a whole different ballgame. But for the manufacturing of viral vectors, the US doesn't require a GMP plasmid. Several European countries don't require GMP plasmid, but if you talk to the Italians, they will require it for every phase of clinical investigation. So really, the answer is it depends on where you go, and it also ultimately depends on whether you are manufacturing for early phase clinical trials or for a commercial product, because although it may not be required by the FDA, the sponsor may want to go for GMP quality components because it is part of their risk assessment, and the more control, the better many times it is for them. So.
Great, thanks, Han. Boris, question for you. How is the titer of integrase-deficient lentiviral vector in comparison to integrase-competent vectors?
Well, thank you so much for this question. Excellent question.
Generally, titers of integrase deficient lentiviral vectors quite comparable to titers obtained from integrase competent lentiviral vectors. Again, in terms of expression, there are differences, and usually integrase deficient lentiviral vectors show lower expression levels. But again, I want to emphasize that in terms of titer is quite comparable between IDLV and the ICLV, and thank you so much for this question.
Great, thanks, Boris. Miguel, we have a question for you. One of the audience members asked, is Transit IT Lenti intended for research use only, and does Mirus plan on offering reagents for commercial use?
Yes, great question, thank you. The Transit Lenti transfection reagent is for research use only. However, we are offering the new reagent, which launches tomorrow, that I mentioned at the end of the presentation, Transit Virus Gen. I would steer people towards that reagent for commercial use, and with this particular reagent, in addition to suspension and adherent lentivirus production, it also supports suspension and adherent AAV production. But for the research use only, researchers that are interested in only generating lentivirus, Transit Lenti is what we recommend for adherent and suspension lentivirus production. Thank you.
Great, thanks, Miguel. Han, we have a question for you. How do you scale up transfection for large-scale manufacturing?
You know, that is a good point, and that is something that really everybody struggles with. As I mentioned during the presentation, that the transfection procedure doesn't really scale up that well, and the reason for that is in the mixing and the fact that time and temperature and how fast you mix all play into the quality of the transfection reagent. One way that we have solved it is we actually defined a procedure for our batch size, and then if we needed to manufacture more material, instead of doing a true scale-up of the transfection procedure, we basically kept the transfection procedure the same and manufactured additional batches. And you will see that if you compare a small-scale transfection in a 10 centimeter plate, where you not only mix your reagents in a small tube and drip that onto your cells, the dynamic is completely different if you go for large scale. And so frequently we do not see that same dynamic being repeated between the small scale and large scale, and then titers start to drop, and everybody shrugs their shoulders in a sense that, you know, that's normal when you scale up. Well, in a way it is normal when you scale up. Ideally you would want to control all the parameters that are needed for the process. Unfortunately, transfection is one of those steps that once you get it right at a certain scale, you may want to stick exactly with that scale. And if you need to produce more, you just repeat the same procedure multiple times to create multiple batches. So I hope that answers the question. Scale up is difficult, and for larger scales, it is generally always a problem. But if you validate your procedure, you make sure it's repeatable, and you stick with that, and you don't make any changes, then it is certainly doable.
And stick with us. We have another question for you. The audience member would like to know, what is the largest scale for suspension culture that was demonstrated for AAV viruses?
You know, that's a moving target. And people are working on larger and larger scales. The scale that I am familiar with, which is a baculovirus manufacturing, is a 500-liter scale. That was the scale that's been demonstrated. But it wouldn't surprise me if people are working on larger scales, very similar to what's being done in the protein manufacturing industry. But I'm personally not aware of it. But the short answer would be, I know of 500 liters at this time.
All right, Han, and one last question for our webinar for you as well. So the audience member would like to know or ask us that they say, as I understand, there has not been much success with producing lentiviral cell lines because of toxicity with VSVG. Has this problem been overcome?
So yes, the problem has been overcome, and as I indicated, this has been done using induction of the gene of the VSVG, induction of the expression when the virus needs to be generated. The group in Memphis has done a lot of work on this, and this is already several years ago. There's a publication by Thrum and colleagues that includes Harry Malik and Brian Sorrentino from the Memphis group. This is a 2009 publication in Blood, and they describe the generation of a number of stable producer cell lines that have VSVG in them, and their concept has been moved into large-scale GMP manufacturing. So yeah, the problem has.
All right, thank you, Han. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site at www.genengnews.com for up to 6 months. So if you missed any parts of it, you can watch it again, or you can feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Miguel, Han, and Boris for their informative presentations, and I'd like to thank the audience for all their great questions. And a very special thanks to Mirus for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.